Good morning, everybody. We're going to get started. I would much rather just keep basking in the awesomeness of the crowd and walk around and sort of keep the surreal feeling of having all of you here today in Toronto for the meeting. I'm going to start with a land acknowledgement first. So we acknowledge that the events and meetings we host take place on land traditionally inhabited and cared for by First Nations. Toronto is the traditional territory of many First Nations, including the Mississaugas of the Credit, Anishtabek, the Chippewa, the Haudenosaunee, and the Wendat people. Okay, so it's really an incredible honor and pleasure to have you all here today at a meeting that's been really a staple for me when I first started the lab. So a big welcome from the organizing team. I have to admit, kudos to Homera for first suggesting that we have the meeting here in Toronto, and then the rest of the team for really, taking it to the next level. So I'm really grateful for everybody on this slide. Grateful for you to be for you being here, and grateful to host this meeting, which has been important for me in my scientific journey. A couple of things that have changed a little bit. So we took a bit of creative license. I'm going to give you a bit of history on the next slide, but we kind of changed the name a little bit to the Human Neuron to Brain Meeting. It's a little bit with a feeling which maybe we don't all share, but I definitely feel, which is increasingly, as we sort of study cells and circuits of the human brain, we really want to translate that to the brain, the functioning brain, and I think we're getting to a point with the technologies that are available to us, the neurotechnologies, that we can start to sort of make that leap from our in vitro work to the functioning and maybe disordered brain as well. The other unique thing is that this is the first time that this meeting is hosted in North America, so very grateful that many of you came a lot further than you'd have to if you're going to Amsterdam, where this really started. And that's really why, for me, I'm very honored to have and host this meeting here. Way back when in 2014, the lab had just started, just published first paper in Human Slices, and out of nowhere, Hal Mansveld there emails me and is like, hey, do you want to come? And I thought, wow, this is crazy, like I'm nobody in the scientific space, and he's inviting me to come, and I really felt really included in the community, and especially at a young time in my career. And in part, that's why as a committee, we've maintained that philosophy here, which is to invite people who are early on in their career who are making significant contributions to the human neuroscience field. So that's an important value, but the other really fun thing that I derived from the meeting was the sense of community, and I hope that you'll all sort of take time to get to know people you don't know here at the meeting, and it's an incredible group of really incredible acumen in this room here. So very grateful for that. Okay, so a lot's happened since 2014. Well, we had the meeting in Amsterdam, and whether it's a correlation or causation, it kicked off a lot of things that have happened since that time in human neuroscience. I know there's a lot of stuff missing, so just it's really just to sort of emphasize just how much transformative work has happened. In 2013, the Human Brain Project, which, correct me if I'm wrong, Natalia, or how that's really what kicked off that initial meeting in Amsterdam, was the funding for that. And then since that time, we've seen sort of the powerhouse of the Allen Institute really transform neurosciences as we think of industrialize the kind of things that had been done in a bit of a hodgepodge around the world, which has really given us a really strong basis for the science that we do. We see a lot of new techniques emerging. We saw the emergence of patch-seq, which was really important for the phenotypic characterization of human neurons. And then, of course, as Ed is here, just through his leadership, really generating really large consortia around sort of tackling really large-scale and important projects that have created an undergird for the science that hopefully we'll do in the next 12 years, 10 years. I think one thing too, this meeting's a little bit different in that we've sort of moved a bit into in vivo technologies. So we've have a little bit of a new component for the meeting this year because of, again, I think this desire to translate to the functioning human brain, and we're seeing the technologies enabling for that. So we have really key people, and all of you have been really part of this this last 12 years. And so we're going to end the meeting with a bit of a panel discussion, but really everybody is invited for that panel, is to maybe think about what the next 10 years looks like and what you want to accomplish and what we can do collectively together. So, so the next 2 days are really a sort of a celebration of all the work that you guys have done, and then also to look forward in time. Okay, with that, looking forward, very, very grateful that Ed is here with his other big parts of his team here to kick us off. He's, of course, no stranger to any of us in the human space, and so maybe a little bit different again. We'll have we're starting with a keynote for this meeting, because I feel, I don't know if everybody shares that with me, but I think we are at a cusp. I don't know how steep that cusp is, but it's a cusp, I think, in human neurosciences, and who better to sort of lay the first, I don't know, what is it, the foundation, or the, I don't know, when you build a fence, the picket, I guess. So Ed, please.
There we go. Well, good morning, everyone. First of all, I'd really like to thank Taufik and the organizers for the opportunity to speak to all of you today. I think it's rare to find an audience that's more enthusiastic about human neuroscience. So I really, really look forward to this meeting and honor for the opportunity. I'd really like to take this opportunity to tell you about some of the work that we've been doing at the Allen Institute and through some large consortia to really try to accelerate progress in human neuroscience. And more, we have been thinking about how to accelerate disease research and translational research as well. And I'd like to just begin by sort of a motivating statement for us recently, which is, why aren't we making faster progress in understanding brain disorders? It's really an astonishing number still. 1 in 3 people worldwide suffer from some form of neurologic condition. And it's sort of depressing, the success rate of things trying to get into the clinic. I think there are many reasons for this. We have a huge knowledge gap. There's a lot that we don't know about human brain structure and function in the healthy brain, much less disease. There's not really a culture of data sharing across many parts of this community, especially on the clinical side. And that leads to consequences. We have poor diagnosis, few treatment options in many cases. And this really is ripe for a change in the model and how we do things. And I think a couple of high-level problems in the space too is that there's not enough investment at the foundational level, and efforts are really fragmented. And in particular, I want to highlight that point, that we have different efforts that are really not connected across the field, and that impedes progress as a whole. But I think we're also at a time for some serious optimism. There's been a lot of progress. There have been some significant foundational investments from the NIH in the US. It gets a lot of bad press these days, but still really our allies in making progress. And I'd like to tell you about some of those today and where I think that this field is going. And in particular, I'm going to be highlighting three areas that we've been involved in. One is to take advantage of new single-cell genomics technologies, single-cell genomics and spatial technologies, which at scale are now producing a really foundational cell reference or cell atlas of the human brain. This is really going from proof of concept to a whole brain atlas in a very short period of time that serves as a foundation for the community to think about. The second is that we can apply that atlas right away and start to use it to understand disease and start to bring the level of cell and circuit resolution to our understanding of disease phenotypes, which I'll illustrate in Alzheimer's disease, but I think is generalizable here as well. And then the third is that this molecular approach to understanding cell types also lets us understand gene regulation, and that leads directly from an understanding of the cell types to the ability to manipulate them and to genetically target them. And so initially, these are foundational tools for neuroscience, but very quickly, this can turn into precision medicine, where you target vulnerable or affected cell populations in disease. So I'll try to illustrate these over the course of the talk. But let me start by just sort of philosophically that, I think we really need a unifying framework for neuroscience that allows us to aggregate information for the information that's generated by all of you to contribute to a broader understanding. And I think of this really as sort of a combination of a spatial framework, a la Google Earth, for the brain, and a cellular framework now that's allowed by these single-cell genomics technologies. And if we could put these together, we would now have an integrative framework that would really bridge all kinds of different parts of the field. It's a common coordinate framework for us to aggregate information spatially. We can bridge scales from neuroimaging to cellular and histological levels. We can bridge structure and function to some degree, and hopefully bridge research and clinical practice as well. On the cellular side, this is now a really powerful tool that allows you to integrate data across anyone in the world who's doing single-cell genomics work to a common reference framework. This allows you to aggregate data. It's really a human genome-style reference to map to. It allows you to link across species, from human to model organisms, across the lifespan, and it allows you to understand disease at the level of very specific cell populations. And this is really quite feasible now to create such a thing. Thank you. That's not moving. This is really feasible to create such a thing, as I'll illustrate to you, that allows us to then integrate data across many different modalities, from single cell to neuroimaging. We can even imagine that we can start to integrate functional analysis as well from the clinic. And the data sets that can be aggregated now are really primed to start to bring advances in machine learning and AI to bear as well to analyze these data. Now, there are lots of barriers, and I'm going to illustrate over the course of the talk some of the barriers and gaps that we've been trying to tackle with these different projects. On the one hand, these are things that really can't be tackled alone. These have to be big collaborative efforts. And we've been fortunate to have the support of the government to be able to build consortia to tackle some of these big projects. We need access to high-quality human brain tissues. And this turns out to be a really non-trivial thing. This is true for both postmortem tissues and living tissues, of course. And as you start to study disease, we have to deal with geographically distributed tissues and how to standardize across sites. We need scalable technologies. The brain is huge, it's complex, and we need scalable technologies that are up to the task of actually characterizing that complexity. We'd like to be able to link macro and micro scales from neuroimaging to histological level. I'll illustrate this a bit. And ultimately to link structure and function and to understand functional deficits in disease. Once you get into disease, of course, we also have issues of human variation and population heterogeneity and disease heterogeneity that become very complicating factors for understanding. But most of all, I think we need to understand disease at the level of brain architecture. And by that, I mean the cell types and circuits that make up the brain, and not think of treating disease as only going after a gene. We can go after the cell types and the circuits that are perturbed or affected in disease as a target. So I'm going to use two consortium projects that I lead that are ongoing right now and sort of weave them together a little bit to illustrate some of these points and the things that we're doing. And I really want to emphasize how collaborative this is and that I'm representing a large group of people. Some of them are in the room here, in particular one table over here on the right with many of my colleagues, that have sort of joined together to try to tackle some of these really complex and challenging multidisciplinary problems. One of these projects is funded by the NIH Brain Initiative. And our particular project in this is called the Human and Mammalian Brain Atlas Project, or HNBA. The other is a U19 project funded by the National Institute on Aging, which is called the Seattle Alzheimer's Disease Brain Cell Atlas, or CAD. Both of these are consortium projects meant to bring together the right skills to really build a foundational atlas and then apply that atlas to understand disease. Let me start with the brain preparation, and post-mortem brain in particular. So I'm going to spend a lot of the first part of this talk talking about post-mortem brain. Really getting access to high-quality tissues is essential for this, but if you can solve that problem, all of the modern molecular methods work on these tissues, and you can very quickly apply them to post-mortem brain. We started this many years ago when we sort of realized that there was a gap in how brains were prepared, and worked with a colleague at the University of Washington named Dirk Keen. It's a neuropathologist who was willing to implement new methods. There's nothing really particularly fancy about this. If you work in model systems, you need to adequately prepare the brain, slab it thinly so you can preserve the anatomy, freeze it properly, vacuum seal it, photo document. But you put those things together and you end up with a system that allows you to prepare whole brains in ways that are amenable for all of our modern methods. Those tools can then be combined with standard reference atlases to begin to pin things into 3D coordinate spaces. And for quite some years, we've been developing traditional plate-based reference atlases of the human brain, annotating or parcellating 3D atlases with that structural nomenclature and parcellation. In this case, this is the MNI 152 atlas in Simon, Canada. And these can be used to pin. And so that's actually quite frequent now in the living tissue studies as well, to pin your samples into a 3D coordinate space that you're able to use for functional studies. This works very well for a coarse registration or pinning of postmortem brain samples. But we're really trying to push beyond that now. And the Brain Initiative efforts have led to another wave of innovation here to try to map a postmortem extracted brain into a common coordinate framework that's based on neuroimaging. And that requires you to go back upstream in the process. We have to actually get a surface scan, or even better, an MR of that brain, postmortem brain, so that we can then map that into an MRI reference framework and then map slabs that came from it into its own volume. This is possible to do through the combination of a surface scanner that you can buy on Amazon, or better, a low Tesla MR, like a hyperfine MR, that you can do on a postmortem brain, either in cranio or ex cranio. And that lets you get a quick scan of that particular brain. You can use machine learning approaches to sort of upsample that to look much like a 3T clinical scan, and that can be mapped into a reference framework. On the other hand, the slabs then, if they're done extremely uniformly and done with photo documentation, can then be aggregated into a volume. And the outcome of this is that we can map any parcellation from a reference atlas, such as the Human Connectome Project, or information from that particular individual's brain onto the slabs and use that as a guide to do molecular sampling. And just to give you a quick little look at this, this is the work of Eugenio Iglesias. Here is on the left-hand side is a 0.07 Tesla hyperfine scan, which has then been turned into a synthetic T1 that looks a lot like a clinical scan, and then can be parcellated with the Human Connectome Project parcellations. That then can be projected down onto the actual physical slabs of the brain. And so we're now getting into a place where every one of these brains can be mapped into that space. We can really use the MR as the way to localize anatomy more precisely and then sample. And conversely, any sample ends up back into a common coordinate framework. Now, we've been trying to push this even to see if we can link structure and function and see if there's a relationship between the functional architecture of an individual's brain, which can't be predicted from a probabilistic atlas, and the cellular molecular architecture by trying to recruit individuals in end-of-life situations to get in a scanner, have a battery of stimuli to map out their functional architecture, and then donate their brain so that we can try to do a correlation to all the other things that we can measure. This is sort of a particularly difficult thing in the US. We've been trying to do this with hospice care. I think this is actually something that could be more easily done in Canada with things like medical aid in dying, for example. But at any rate, this is very generalizable. You can imagine in the future we'll have scans on individuals who then donate their brains, and we can look at the correlation between pathology you can see in neuroimaging and what's happening at the cellular histological level. So this creates something of the spatial framework. Let me shift gears now to the cell atlasing component of this. And here we have had a real transformation in the field through the increasing availability of high-throughput, scalable, single-cell genomics technology platforms. In particular, 10x Genomics has come along with an extremely scalable set of technologies for single-cell RNA-seq, for ATAC-seq, or for the combination thereof. And this lets us tackle cellular diversity in extremely complex nervous system tissues. And so we can take brain regions, we can dissociate them. In the case of human brain, we can do this with nuclei. You can profile them to measure many thousands of genes in each cell. This can be used then to cluster and to derive a cellular taxonomy. That taxonomy turns out to be extremely useful because it's now a framework for aggregating information. And we can begin to then understand the properties of those molecular types of cells. And sort of fast forward, this molecular taxonomy turns out to be extremely well correlated with many other cellular phenotypes and developmental origins of cells, for example. It also drives to the creation of genetic tools. Some years ago, when the Brain Initiative was first starting, in the first phase of this called the Brain Initiative Cell Census Network, the entire consortium worked on one brain region, the primary motor cortex, and really established the paradigm for how this cell atlasing could be done. Really deep sequencing of that particular brain region leads to a quantitative taxonomy that can be represented as a hierarchical cellular taxonomy from class to subclass to type. You have the cell types, you have their gene expression profiles that give rise to their properties, you have epigenetic profiles. With the spatial techniques, now you get their spatial organization, tissue microorganization, and proportions. You can start to layer on different pieces of information. So by parallel to the human genome, once you can define the genes, you can start to layer more and more and more information on this. It becomes a very powerful framework. And it also can be used to quantitatively map across species. Fast forward 2 years, and we already had a complete mouse brain with these techniques, with both single cell and spatial, and the first draft of a human brain. Incredibly scalable methods. What this revealed is the complexity of the system. In the mouse, there are over 5,000 discriminable types of cells, most of them neurons. In the human, the first draft of about 100 brain regions got to about 3,000 types of cells. So it's just an exceptionally complex system. I'm part of the Human Cell Atlas that's doing the similar cell atlasing in different organs across the body. The brain is more complex than the rest of the body combined. The success of this in the human led the NIH to propose and fund a second phase of this called the Brain Initiative Cell Atlas Network, or BICAN, which is really trying to go comprehensive on the human and non-human primate. And furthermore, to do this across the lifespan and across populations through a coordinated set of projects. And this effort is really now trying to turn this from a scientific output to a foundational reference that the community can use and really formalize this much more. Our particular component of this, as I mentioned before, is called the HN2B, or Human and Mammalian Brain Atlas Project. And this is really the atlasing project of the whole consortium. We wanted to cover the entire brain with single cell and spatial methods. To do this in a coordinated way between human, macaque, and marmoset that could be linked to the mouse, and to also begin to annotate this with techniques that many of you are using of slice physiology or patch clamp physiology to start to understand the phenotypic properties of cells in this molecular taxonomy. I'm going to illustrate in the first brain region that we tackled how this is going. The consortium, as I mentioned before, for the primary motor cortex has done a similar thing as in the first phase of the BICAN phase of this, to focus on the basal ganglia. And there are many reasons for doing this. When we get into human, there's a lot more emphasis on disease-relevant regions of the brain. It's, of course, associated with many diseases: Parkinson's, Huntington's, Tourette's, also addiction, obsessive-compulsive disorder. Lots of reasons for wanting to study these. And the consortium as a whole put all the collective resources into studying this from the different angles and the different methodologies that the group had to bring to bear on this. It now contains the beginnings of well-annotated, cross-species harmonized common coordinate frameworks, or reference atlases, as I was describing before. This is actually something which has been quite challenging to do because there are many atlases out there that don't use the same terminology. So we're trying to have one that's based on a common definition with common nomenclature that works across species, and to build, use that structural nomenclature then to inform the cellular nomenclature as well. To put a whole lot of work into one slide here, by sampling with single-cell genomics across all 3 species, we can generate an integrated cell atlas of the basal ganglia structures. This covers all of the cell types that make up the basal ganglia. And down to a certain level of resolution, these are clearly homologous across species. And so we can integrate this and create a harmonized atlas. And this is sort of generally true, that down to a certain level of resolution, there is a mammalian blueprint. And we see the same cell populations. But when you get down to the finer levels, you start to see a lot of species specializations. And by no means do I mean that there are no molecular difference or other phenotypic differences. There are lots of them. But there does seem to be this core conserved cell makeup of the brain. In addition to the cell classification, that can then be used to drive spatial transcriptomics to map the distribution of these cells across the topography of these structures. Merfish or Xenium are the current commercial platforms for doing this. And so these can then be used to map this and get a spatial map of these cell types. And then a significant patch-seq effort in macaque in particular has now annotated what the morphoelectric properties of these cells are as well. So it really creates a powerful reference. And we've built tools for people to map their own data into it and transfer labels onto their data sets. I want to mention just a little bit more on a few of these topics. We have invested for many years being able to work with human neurosurgical resections and do various types of techniques, most notably patch-seq. This has really been a fruitful collaboration with many of our neurosurgeons in the Seattle area. This was not possible to do for the basal ganglia because we simply can't get access to tissue. Maybe some of you will find ways to do that, but at the moment we have to go for a proxy, which is the non-human primate, where we have access to tissues from the Washington National Primate Research Center in Seattle that allowed Brian Kalmbach, Jonathan Ting, Brian Lee, Rachel Daly, who are all here at the meeting, and others to begin systematically characterizing the properties of those cells. The spatial transcriptomics allows us to really look at the distribution of these cells and local tissue architecture. And of course, this recapitulates some of the major divisions of the striatum, but there are also many highly variable domains, such as the striosomes. There's core shell organization. There are gradients across the striatum in different dimensions. So we really can get a full picture with this of the architecture of the system. I want to highlight here one of the challenges that this presented to do the spatial work. The commercial platforms for doing spatial transcriptomics are somewhere between a 1 by 1 centimeter and 1 by 2 centimeter, which is fabulous if you study a mouse or you look at a biopsy. And it's really a problem when you get to very large brains because it's simply not enough to try to cover the topography of those regions. So the commercial platforms allowed us to do more or less whole marmoset sections. To get macaque striatum, you had to tile it into a couple of tiles. For human, we had to tile it into like 9 tiles to get a single plane of section through just the striatum. So you can see how insufficient these methods are now for if you want to do a map of the entire brain or really get a look across the whole brain in disease, for example. And that's really led us to think about new technologies that are scalable to be able to look at whole brain, or whole hemispheres of the brain, I should say, to do this. And of course, there are methods out there that have been piloted, but they haven't really been scaled yet for doing histology, for doing spatial transcriptomics, now even moving into proteomics. And with the light microscopy versions of connectomics like Lein, you can start to imagine that one could scale up to do actual connectomics. And just to show you that this is kind of not crazy to think about doing this, we've been collaborating with a chemist at UCSD named Bogdan Bintu, who trained with Xiaowei Shuang, who developed MERFISH, to work on a larger format version of MERFISH, which he calls MERFISH Plus. This moves from a more or less 1x1 to something about 10 times the size of that. And in this slide, you can just see that he used this not for a large format section, but to put a whole mouse experiment on a single slide to look across the mouse brain. This can be applied to look at human brain now, a much larger chunk of human brain. And there's no physical reason that this can't be scaled more. And so we're actually working with him to develop the next version of this that could go to whole hemisphere size with full spatial transcriptomics. And now we're talking about, look at 1,000 genes across a whole human section. So this is where I think we need to go. We need to be taking a much broader look at the human brain. It's really insufficient when you think about disease that we're myopic and we look at this tiny little bit of the brain. And especially when you compare it to what you can do in mouse now, where you can do whole brain scanning, we're really missing the picture if we can't look more broadly. All these data from BICAN are made public, and this is really part of the mantra that we've professed for a long time, and NIH has really bought into now that these data should be made publicly available immediately. And furthermore, we should put a lot of effort into creating resources to make them usable for the community. And so all of these data that I've just described are available. And this was just the first region. So in the time that it takes to annotate one region, the rest of the brain is almost done now. So, so we're now in a process of data annotation. And in the next 2 years, you can expect a fully annotated version of the entire human brain mapped across species. And so I just really wanted to cap this part of the talk by saying that these really are sort of genome-style references, and the robustness of these techniques allows us to treat it somewhat like the genome, where we can now start to have really formalized versions and iterative versions of these cell taxonomies and build cell ontologies and formal nomenclature systems around these that work across species. We can have tools to be able to visualize these data, to mine them, to use them more and more for AI types of applications, and tools for people to be able to map their own data and transfer those labels. So this is very much like BLAST for the genome. So it's transformative when you can map to the genome and your own sequence could get annotated. You can do this now for cells as well. And then finally, the knowledge just starts to build. And so as you map into this, you can take advantage of this incredible growing knowledge base about everything we know about these cells, the molecular underpinnings of these cells. Okay, so in the next part, I'm now going to show how this can immediately be used to study disease. And this is with the C.E.T.S project that I mentioned at the beginning, where our idea was, now that we can map at this very fine resolution and understand at this fine resolution, we can use this now to understand disease progression and whether there might be vulnerable cell populations and whether there might be a stereotypy to this procedure. We can map disease trajectories and understand it at this very fine level of cell types and the molecular pathways that are perturbed within them. And this might actually lead to new ideas of where we should go after targets, not just going after amyloid and tau, but go after the cells that are affected. The overall strategy in a nutshell is to combine quantitative neuropathology with single-cell genomics. And we can map disease trajectories as increasing severity of pathology. This can be modeled. We can build pseudo-progression of diseases. And then to profile disease tissues across cohorts. So we now have to move from a few cherry-picked brains to large cohorts of individuals and map to the cellular reference. So we can actually understand and interpret at very fine levels of resolution. And now we can start to look for cellular effects. And this can be at a gross level, this can be proportional differences that happen over the course of disease. It can be, of course, molecular differences that happen as well. To do this, we have to get access to cohorts of individuals. In this first phase of the C.E.T.S project, this was entirely a Seattle-based project. We had access to 2 longitudinal cohorts that are happening in Seattle area. One is an Alzheimer's disease research center. This is one of the national centers where people come in when they're symptomatic. This is a treatment center. And then they're tracked. They have cognitive testing over the course of their life, and most of these end up donating their brains. The other is a population study. This is an aging and dementia study run by Kaiser Permanente called Adult Changes in Thought, or ACT. And in this case, you can't be symptomatic at age 65 or older when you enroll, and then they track you for the rest of your life. So these two together allowed us to build a cohort that spanned the full spectrum of Alzheimer's pathology, from no pathology to very high levels of pathology, which is really key for the idea of sort of building a trajectory of disease. This spanned the spectrum of known Alzheimer's pathology, both amyloid measured by Thal, or hyperphosphorylated tau measured by Brock. So we really do seem to have covered the spectrum of disease, of progression from an Alzheimer's pathology perspective. I will say one of the things that we quickly learned entering this field is that co-pathologies are the norm, not the exception. And so actually there's a huge amount of heterogeneity here with other co-pathologies. And mixed pathologies is really common these days. And I think that creates an even bigger problem of needing to scale to much larger cohorts that can span that heterogeneity. Something I'll just mention here right away is that the practices for brain collection are really variable across the country and across the world. And frankly, most of it's not very good. There's a lot of tissue of sort of highly variable quality. And so we've been trying to disseminate the methods that we've developed across many different sites, both in the brain initiative work and the C.E.T.S work. And this has now been adopted at about a dozen sites. So it is a technique that can be replicated, and that allows us to then really control the upfront part and reduce the variability associated with that. But I will say that that's mostly worked when you can fund, when you can supplement, rather than just trying to convince people to do it. So I think there's a lot of work that could be done to make this easier and make sort of turnkey solutions for being able to adopt these methods. The idea here was to really follow the pathology. And we took sort of 2 different approaches to this. One is to look in a particular brain region and look at cellular effects as a function of increasing pathology. And the other is to follow the progression of pathology across the brain, and in particular tau pathology, which begins in the entorhinal cortex, moves through the hippocampus, up the temporal lobe, and then out into the rest of the cortex. Using the pathological measurements and ML approaches to extract features, we're able to build pseudo-progressions of disease progression. And this really seems to largely be driven by Alzheimer's pathology, amyloid and tau, but also is actually seen with other phenotypes. We had actually included NuN as a neuronal marker as a control, which turned out to be anything but a control. It really decreases as a function of increasing pathology, and is probably a broad neuronal effect that is as a disease effect. But this correlates very strongly with decreasing cognitive capabilities and more qualitative metrics of Alzheimer's progression. In order to really do this well and do this across regions, we have to build these cell taxonomies. We actually got a little ahead of the brain initiative work for this. We had to create a cross-regional taxonomy that moved from the hippocampus and entorhinal cortex and across the neocortex. Fortunately, there is a pretty canonical organization, so you can map cell types across these regions. For a lot of cell types, like interneurons and non-neuronal cells, these are actually well conserved across all these regions. There are significant differences in the excitatory neurons and in very specialized regions like primary visual cortex. But we are able to create this integrated cell taxonomy, and that now lets you look for cellular effects and how they compare across brain regions. So I'm going to try to now explain some of the sort of core first-order results of this to give you a flavor of what we can learn from this. We can now look for changes across about 139 different types of cells just in one region of the cortex. And we can look for proportional differences along the pseudoprogression that represent either a decrease, as you would expect for neurons, neurodegeneration, you only lose cells, or an increase in certain non-neuronal states. There are well-known disease-associated microglial states and astrocytic states, for example. And so what I'm showing you here are the proportional changes just in the middle temporal gyrus, where on the right-hand side in the red box here are the non-neuronal populations, and there are actually a few of these that go up, as expected, the microglial and astrocytic states. In the excitatory neurons, most of the action is in the upper layer, supragranular layer 2-3, intrathelencephalic projecting types, so cortical-cortical, corticostriatal. And then significantly broader effects in the GABAergic populations, both of all the major subclasses. But I want to highlight how much specificity there is here as well. So it's not all cell types that are affected. In fact, there's very little happening in the deeper layers of the cortex. So it's both specific and yet kind of broad as well. Now, if we look across regions, I'm going to sort of march you across these regions now. So moving from 1 to 10 regions, starting with the middle temporal gyrus, and going back towards where tau originates. So inferior temporal, medial entorhinal, lateral entorhinal, and hippocampus. And it's incredibly stereotyped across regions, the pattern of cellular vulnerability. If we move from MTG out further into the cortex, superior temporal, frontoinsular, dorsolateral prefrontal, angular, and even primary visual cortex, it's just incredibly stereotyped across the cortex. So there seems to be a canonical pattern of cell vulnerability. That's not to say that there aren't regional specializations as well, or regional effects as well. And if I could just draw your attention to one of these at the very bottom of this plot, in the primary visual cortex. V1, of course, you all know, is highly specialized. It has a highly expanded layer 4. This is reflected in an increased diversity of the molecular types in that region as well. And these actually seem to be quite vulnerable as well. And we see significant losses of the input layer of cortex. So I think this is quite a surprise to see this sort of thing, because usually you think of this as a relatively spared area. But actually there's a literature on visual effects in Alzheimer's disease that this might actually relate to. So I think we're starting to get at the substrate of cognitive effects in Alzheimer's disease, and this is something that can be applied very broadly. I want to make a couple other points here. The first is that with this reference, any other data set can also be mapped into this reference. So we've been mapping all the other available single-cell data sets against this same reference, and we're able to replicate some of those effects, in particular microglial effects, some of the GABAergic interneurons, the layer 2-3 excitatory neurons, for example. It's also highlighting that most groups actually don't really sample enough to fully take advantage of this reference. If you don't sample deeply enough, you don't have the power to look at very fine types of cells that we can with the deeper sampling. The other is that we've tried to replicate this in data available from mouse models of Alzheimer's disease. And while all of them seem to replicate the inflammatory microglial phenotype, none of them so far replicate this selective neuronal loss. So we have a whole field studying something which really doesn't replicate many of the primary effects of AD in the human brain. And I think this really highlights the need to study human and the need to find model systems that actually replicate some of the incredible diversity of the human brain that may not be seen here. Things get even more interesting when we start to look at trajectories. So as one might expect from the deep literature, there are early and profound effects on the non-neuronal cell populations. There are early and progressive effects on the microglia and the astrocytes. When we look at neurons, on the other hand, we see this loss of the excitatory neurons very late, but it turns out that very early on, there's a loss of the somatostatin-positive neurons, and that seems to be the earliest neuronal effect. This is replicated across different regions of the cortex as well, that this somatostatin loss early it seems to be a common effect. We've now been able to look even more broadly and tried to look at not just the regional pathology, but the global pathology, and start to look at the relative timing of effects across regions. And this plot here is an attempt to show this, where now we have a global pseudoprogression that's represented on the y-axis here. And you can see which effects are happening early and which are happening late. And in particular, I want to highlight on the left-hand side now are these somatostatin cells. Most of these effects are very early in disease progression. So this is before there's much pathology even. We're already seeing the loss of these cells at the same time that we're seeing a loss where we would expect to see changes such as the CA1 neurons in the hippocampus. Many of the later effects, like other regions of the hippocampus, happen a bit later in the progression, and many of the other cortical effects actually happen much, much later in this disease progression. So we're starting to get this sort of progression of what cells are affected and at what point in disease. And then finally, we can take advantage of the information gleaned from the spatial analysis and from the patch-seq that's done on neurotypical, well, not neurotypical, but from neurosurgically resected specimens, I should say, to understand exactly what we're talking about here. And it turns out that the vulnerable somatostatin-positive cells are located in the supragranular layers, the upper layers of the cortex. And of course, these cells are well known to be extremely diverse, but they're very heterogeneous, but also include the famous double bouquet cells as well as some of the most vulnerable cell populations early in disease. So here we are. We've gotten to a level of understanding some of the cell populations that are affected at the finest granularity that we would have in any model system that we can now understand in human. And you can start to now think about what this might mean for circuitry. And from my perspective, this just turned Alzheimer's into a circuit disease. And we have loss of very specific circuit elements. We can now think of it this way. We can start to think about what that might mean. We can start to model it. And this might lead to an understanding of how the sort of substrate behind some well-known phenotypes of AD, such as early hyperexcitability or even seizures that one sees in Alzheimer's disease. Maybe these are also a sequence of events. You lose certain elements that are trophically connected to others, you might actually see loss of others. And something that's quite clear from these data is that most of the vulnerable cell populations are commingled with one another and therefore likely connected to one another. So at any rate, you can start to think about various new ways of thinking about the disease as a circuit, and the cells that may become now sort of a new place to shine the spotlight on, or even target as a therapeutic. So I'd like to have the last bit of the talk on that topic, because what I think will be one of the more transformative elements out of all of the cell atlasing work is the ability to manipulate. And I don't think we really fully appreciated this when we began this work, but the cell atlasing, and in particular the epigenetic data, allows us to identify short regulatory sequences or enhancers that are sufficient to drive cell-selective expression of a transgene when put in the context of a virus. And we can mine these cell atlases for cell-selective peaks that are open chromatin in particular cell populations. When we screen these in mouse, it turns out that many of these are in fact functional enhancers and give predicted cell-selective expression. And many of these then are also conserved across species. So we can move from screening them in mouse to monkey, and I'd say probably the majority of these, at least at this sort of intermediate subclass level, show pretty conserved expression from mouse to a monkey, and therefore likely to a human. And so this is really a path to being able to target particular cell populations that may be affected in disease. Just to extremely rapidly show this, we can really target just about any kind of cell in the brain now. Jonathan Ting has a remarkable program of being able to identify regulatory sequences for many cell populations. These work in mouse. Many of these have been tested in macaque and really can target almost any cell population. They can be combined with any kind of cargo. They can be dramatically enhanced through bashing and concatemerization, for example, and then they can be combined together. You can have multi-specificity single vectors as well. So these are really powerful tools to be able to start thinking about targeting relevant cell populations in disease. And for a number of years now, we've been exploring this idea that cell-selective therapies could be achieved with this approach in childhood epilepsies. Our colleague Boaz Levy at the Institute has been driving an effort in Dravet syndrome as one proof of concept here, where this is an intractable epilepsy. It has sudden infant death. It has various seizures. There are no treatments for this at this point in time. And it's a good target for this kind of approach because we know the gene that's defective. If we could replace that gene, we know the cell population that's particularly affected are GABAergic interneurons. So this really allowed us to put these pieces together to use a highly optimized GABAergic enhancer to drive transgene expression, to develop a way to be able to augment expression of the SCN1A gene that's affected in this disease. This was a little bit complicated because it's too long, too big for an AAV, so we had to come up with a two-part system, a split intine system, to be able to reconstitute this, but this works, at least in a mouse. And when you apply this, we can get some pretty remarkable phenotypic rescue of these cells, of these mice. So for example, the mice recapitulate an infant mortality phenotype. This is completely rescued, really 100% rescue over more than a year. If we apply this to GABAergic cells, serious rescue of febrile seizures and rescue of various spontaneous epileptic symptoms that are seen in these mice as well. Interictal spikes, myoclonic, generalized tonic-clonic. So it can really get quite remarkable rescue. And cell specificity matters. So if we do the same thing, but we apply it to all neurons, it actually worsens these phenotypes. Many more of these mice display an infant mortality at the dose that's necessary to rescue seizure phenotypes. So I think that this may become a running theme for a lot of therapies in the future, that we need to be selective. If you have non-specific targeting, you may get all kinds of off-target effects, and maybe we can improve this by this selective targeting. Just to give you a few more teasers of what we might be able to do with this, in ALS, the motor neurons are vulnerable. We can target both the upper and the lower motor neurons and combine them. So this could be a way to try to target vulnerable cell populations to prevent their death, for example, in ALS. I know there's some interest in neuromodulation here, so I thought I would show a beautiful slide from Jonathan here demonstrating that we can target the various kinds of dopaminergic neurons or serotonergic neurons or noradrenergic neurons or subpopulations of them as well. So there are many, many possibilities for being able to target the relevant populations for different kinds of diseases. We do have to solve the delivery problem, and I think this is a problem for the entire field of how are we going to get brain-wide biodelivery. The race is on, of course, to get blood-brain barrier crossing capsids that work in humans. Again, showing some work from Jonathan here on a capsid from a company called Dino that purports to be a BBB crossing capsid. In fact, this works quite well, at least with a broad synapsin type of promoter to give uniform, broad biodistribution across the primate brain with an IV injection. So I don't think that this is necessarily the solution for this particular vector, but it really highlights that the tools are coming to be able to solve the biodistribution problem. If we can do that, that's combinable with cell specificity and any molecular target for a particular disease. It opens up a whole new suite of possibilities. So I just have a couple slides to illustrate where we're going now in the future. We've really been on a path to imagine where we could go in the future and how we could try to build on these tools for a much broader impact program on human brain health and disease in particular. And really have been thinking of, we need to put things together. And there are all these different siloed parts of the field. Even what I've described here, every part is on a different disease. They're not really connected to one another. But what if we could put all these things together and take a much more integrated approach to the discovery side, where now we can apply these types of tools very broadly across much more heterogeneous populations and start to also connect it more to clinical measurements as well, and connect the longitudinal studies with postmortem brain to try to get at disease mechanism, and then follow that through with a translational arm to take advantage of this cell specificity and think of this sort of in an end-to-end way. I want to highlight one other component I haven't talked about yet, but I think this audience would care about. That, another transformative element in human neuroscience is now the application of high-density silicon microelectrodes like neuropixels in the clinic. And that offers another scalable, high-throughput approach to generate very large data sets that can be used to start to understand human brain function, but also the opportunity to connect those spikes with the cell types of origin and to think about what cells are giving rise to different properties and how they're affected in disease. And this will necessitate a modeling sort of approach in the middle as well to have this iterative loop and to think about how to interpret these things we're getting from the postmortem brain and how to predict what we might be able to do with them. So in a nutshell, we're really imagining how we can bring these scalable technologies to bear on understanding disease progression, couple these with a major modeling effort, and then couple those with a translational effort that can really try to see whether we could advance some of these to the clinic. And I want to highlight how incredibly generalizable all of these tools are. We could think of doing this across a whole variety of diseases with similar effect. Alzheimer's, when we started, there was a huge field of Alzheimer's and many, many people looking at it. But if you can bring a more systematic effort to it, you can learn new things with new tools. So I think there's a lot of opportunity here. But this can't be done alone. And I want to highlight this, that it's going to take a huge global effort to actually apply this and move the needle on this. And we've had a taste of this with what we can do with the project so far, but I think there's just enormous opportunity to partner and to collaborate both among scientists, but also with government, philanthropic organizations, disease foundations, tissue providers, and technology providers now in the AI space. And I hope that we have the chance to work with many of you on that. And with that, let me just end and thank the many, many people. When I've been describing this as we, I mean this in the collective we sense. I've represented the work of an enormous number of people, both at the Allen Institute and our many collaborators, and also our funding organizations, the NIA, NIMH, and the Brain Initiative. Thank you very much.